Great. Good afternoon, and thank you all for joining us. Today is the last seminar in our Spring Core Facility Seminar Series. I am Lisa Korpieski, the Director of Communications and Marketing here at the Institute for Applied Life Sciences. These seminars are intended to provide awareness to the great work being done in the cores and give you all a chance to interact with both the facility team and your peers actively using the resources. Just a few quick housekeeping items. The seminar is being recorded, as you just heard. All replays of the seminars in this series can be found on our website, and I will put a link in the chat. We will save the Q&A until the end. During that time, I welcome you to unmute yourself and ask your questions. Today, we are hosting the Animal Models Facility with Director Wei Cui. Dr. Cui is also an Extension Assistant Professor in the Veterinary and Animal Sciences Department. He will tell us about the transgenic, gene targeting, and other services available in his facility. Wei?
Yeah. Thank you, Lisa. Hello, everyone. Welcome. This is Wei from Animal Models Core. So today I will talk about the services we can provide from Animal Models Core in IELTS at UMass Amherst. So we will talk about the methods that we use to generate genetically modified animal models, which involve early embryo manipulation, embryonic stem cells, embryo transfer, and CRISPR-Cas9 genome editing. Before I start, just want to mention that you can find more information under our website. And also I need to mention that for any work with live animals, PIs need to get the approvals first from IACUC before our core can give the service or help. Our core is located on the 5th floor, south part of LSL, and our mice, our animals, are housed on the 1st floor of the same building. And today we will talk about these models and these procedures. So feel free to ask me, but yes, if you can leave a question at the beginning, that's good. Or feel free to stop me during the talk. So the first model we will talk about today is transgenic animal. So what is transgenic animal? For example, you put a human gene into a cow's body or genome. So you want to get a human protein from the cow's milk. So this is a transgenic cow. So for us, we use mice as the organism. So we can put the transgene, for example, GFP, or whatever you want for your project, for your research project. So we can put this transgene into the mouse genome. Another example is like these fish. You can see different color because they carry different fluorescent proteins. Okay, so how to get these animals? So normally PI, you give us a DNA construct. And normally this construct already has a transgene sequence and also a promoter before that. And sometimes there are also some regulatory sequences in this DNA construct as well. So you give us one DNA construct and we will prepare superovulation, breeding, embryo collection. So eventually this DNA construct will be micro-injected into this one cell stage zygote embryo. So we do per-nuclear injection, meaning this DNA will be injected into this nucleus, which has genome inside. The method is straightforward. The consequence or the process is random. Why? Because for this method, you don't know where this DNA construct will be incorporated into the genome, and also the copy number. It could be one or it could be multiple copies together in the mouse genome. And that's why for the gene expression level, it could be high, it could be low, or it could be silenced. Again, if you just want to get overexpression model and you don't really concern, these limitations, So this model will be your option. And after microinjection embryo transfer, normally we will select several ones as the founder to establish the mouse colonies to get a good one for your project. Okay. So for embryo transfer, depending on the embryo stage, for early stage embryos, normally we need to do surgery. For example, here, embryos are transferred into the oviduct. Or you can do microinjection, the embryo culture to a later stage. For later stage embryos, they can be transferred into uterine horn. Again, here, through surgery. Or for later stage embryos, normally we use non-surgical embryo transfer, which is really similar to human IVF clinics. So embryo or embryos will be transferred into the uterine horn through the cervix. So this method is similar to human patient. And for this method, there's really fast and much less painful for the female recipient mice. So that's the first model, transgenic model. If you want to get overexpression model, but if you want to get a gene or sequence or locus specific model, for example, you wanted to delete one exon, or you wanted to put a point mutation, or you wanted to insert a reporter, for example, all these ones, transgenic model doesn't work. So we need to use a different model. For example, chimera. So in this model, as a PI, you give us the stem cells, embryonic stem cells. So these cells, either you make them in your own lab after electroporation, after selection, or you can purchase them directly from the vendor if they are available. So the point is these stem cells, they have been modified through homologous recombination, which is a process named gene targeting, meaning the gene of your interest have been modified already. So these stem cells will be micro-injected into either blastocyst, which is this one. You see there's a cavity. So these cells will be injected to the blastocyst, or we can also do early stage embryos, for example, 8 to 16 cell stage embryos. Both methods work well. So these stem cells and this recipient embryo, which is here, this recipient embryo. So our goal is these modified ES cells and these recipient embryos, they will work together to produce the progeny. So after microinjection embryo transfer, you get the offspring. So we want to see is there is chimerism or this coat pattern on their coat or skin. So that means these modified stem cells contributed to their, at least the skin part, right? But our final goal is we want these pups in their germline, meaning their ovary or their testes. These modified stem cells also contributed to their germ cells, but we don't know for this generation. So the only thing we can do is wait them to grow up. When they are adult, put wild type mice with these potential founders. Then if in next generation, by doing sequencing, we confirm this modified allele there. So that means there was germline transmission. So indicating these mice, their germline also have their these modified stem cells, okay? But I have to mention for this model, normally these stem cells and these recipient embryos, they are from different mouse strains. So that's why you see different coat color. If you want to get a pure genetic background, so these offspring need to be back cross for several generations to get a pure background. So this takes a long time, around 1 year normally. So that's why this method, sequence, locus, everything specific is accurate. after validation at a stem cell stage. And then we do this chimera, then we do embryo transfer, we do germline transmission, then we do back crossing. This takes really long time. So this is a limitation, takes a longer time. Okay, so the last model we want to talk today is CRISPR-Cas9 genome editing, which belongs to engineered nuclease. You may also hear like zinc finger nuclease or TALEN. So they both also belong to engineered nuclease. But now CRISPR-Cas9 is much more popular, and most labs are just use CRISPR now. Why? Because this system is much easier and more convenient. Why? Because it only has two components. The first one is this, you can see here is this large protein named Cas9. So Cas9 is a nuclease protein, meaning it can make DNA breaks. And if it's wild type, which we normally use, it will make a double-stranded DNA break. So this is Cas9. The other component is this RNA, which is named single guide RNA. It has two parts. The first part has a sequence-specific region. So depending on where you want this guide RNA to bind, so this sequence should be specific, because depending on the sequence, it will be complementary to the target sequence. So they will combine together. After binding here, this guide RNA will guide or will recruit the Cas9 protein coming here to make a DNA break. Okay, so this is this part of guide RNA, really critical for binding, for specificity. The other part of guide RNA is named scaffold, meaning the bone, the main bone, or the main structure of the guide RNA. So this sequence is universal for all species or for most species, meaning you don't need to change this sequence at all for your organism, no matter mice, Drosophila, or whatever. Okay, so guide RNA will find its target sequence and bind here. Then guide RNA will recruit Cas9 protein to come here and make a DNA break. The only requirement is this PAM sequence, which is NGG or CCN in the sequence of your gene. So meaning as long as there's GG or CC in the gene of your interest, you can make a DNA break there. And the DNA break region is upstream of this PAM sequence NGG between the third and the fourth nucleotide. So if you see NGG and the upstream, which is the 5' and you find the third and the fourth nucleotide. So the double-stranded DNA break will be here, okay? So after DNA break, CRISPR-Cas9's job is done. So the following job is for the cell, because DSB is really dangerous. It could lead to lethal phenotype. So the cell has to repair this DSB, double-stranded DNA break. And normally there are two pathways to repair that. The first one, or the dominant one, is non-homologous end joining. So basically it delete more or it insert several nucleotides to make these two broken ends to be glued together. So this is NHEG. So you will see there will be random insertion or deletion, okay? And if this insertion or deletion part is a protein coding region, and if this region is close to the beginning of the protein, this will cause knockout if this insertion or deletion is not a multiple of 3, for example, 5 or 10 base pair, right? Because every 3 base pairs make 1 amino acid. So if it is not a multiple of 3, this will cause frame shift mutation for all the following amino acids. So this will be knockout. The other repair pathway is HDR, homology directed repair. So normally the cell use sister chromatid or homologous chromosome, which shares the identical sequence as this broken one to repair this broken one. But if you give the cell the artificial template, meaning you trick the cell. Why? Because you put exactly the same left arm and right arm there. Then this will trick this broken DNA to think, okay, this is my repair template because left part, right part, exactly the same. So they will use this one to repair this DSB. So what you put in the middle, this sequence of your interest will be knocked into this broken part. So in this repair pathway, the exact sequence of you want will be knocked into this region. So this is knock-in. For knock-in, you can put a point mutation, conditional allele, for example, knock-in LOXPs, or a gene or a reporter, like a GIP, whatever. Okay, so this is knock-in. So normally knock-in efficiency in CRISPR is lower than knockout because it requires both DSB and HDR repair. And this repair mechanism, the success rate or the ratio is lower than NHEJ. Okay, so this is knock-in, especially for large knock-in. The efficiency will be even lower because large knock-in takes or requires longer homology arms, and the HDR repair ratio will be lower. Okay. Okay, so here just want to show you a quick example of making knockout. For example, if we want to knock out beta-actin in mice, okay, so you can use a database to find the mouse species, and you will see there are multiple transcripts for beta-actin gene. And normally, if we talk about knockout, we focus on protein coding ones, meaning we focus on these protein coding transcripts. And our strategy will be, so first we wanted to target a region in the DNA that this region is shared by most or all transcripts. For example, this one, okay, you can see this region of DNA is shared by all of these protein coding mRNAs, meaning these solid colors here, okay? And also depending on the orientation, we can see the arrowhead here. So we know this is a beginning. So for this transcript, this is UTR, no protein here. So we know this is a beginning of the protein. So we want to make a random insertion or deletion to cause knockout at the beginning of the protein part. Okay, so this will be our plan. So we can find the DNA sequence around this region, and then we copy paste into a software, for example, chop. Okay, so you put a sequence, mouse, CRISPR-Cas9 wild type to make a knockout. Then the software will pop out these candidates, which are the target sequences you may use for making the knockout of this gene. Okay, and normally we also do cross-validation, meaning we also use a different software. For example, benchling. So again, do CRISPR guide RNA design, mouse, beta-actin gene. So you will see, again, the first exon 1 is UTR, there's no protein, because the first amino acid starts from this ATG here. So you can copy, so you can select the sequence of this region. Then this software will pop out these target sequences. So you can see on target, off target scores, and also the position, and also the PAM sequence, the scores, everything, okay? In this way, we can compare or validate each other. So we focus on not only the scores, on target, off target. So on target means the specific binding and then DNA break score. Off target means where else this guide RNA may also bind, which will cause off target. Okay, so we consider these scores, and we also think about the position as I explained, because we want this position to be close to the beginning of the protein. So for example, this red one would be a really good one, or this orange one is also a good one. So this will be our top one or top two candidates. So when we know the guide RNA sequence, so we can, so as I highlight here, so this will be this one. Okay, so you can see CGG is a PAM sequence. Upstream between the third and the fourth, meaning between this A and C, the future double-stranded DNA break will be here. So we hope there will be random insertion or deletion, and it is not a multiple of 3, which will cause frame shift mutation. So this will be knockout. When we know the sequence, if you click the off target, you will also see these potential off targets, meaning compared to on target, which is scored 100. Okay, so these are potential off targets. So meaning you can design primers to flag this region if you want to rule out the possibility of off targets. But again, just want to mention, so even this like 1.7 is still much lower than this on target, meaning the possibility of making off-target cutting, this possibility should be really low. And for this one should be even lower. But again, you can design primers to validate your founder or your future mouse lines, okay, just to make sure there's no off-target. So when we know the target sequence, so this is a target sequence, for example, here, okay, so which we get from the softwares. So we use PCR method. Why? Because there's no need to work with plasmid or E. coli bacteria, which is more efficient for us. So we use this method. So how to do that? We order 2 DNA oligos. One is the scaffold, remember, so this part is universal for all single guide RNAs. So this part is fixed. So one time of ordering can be used for many times. So by doing this, we can decrease the service cost. The other DNA oligo will have T7 promoter sequence and the target sequence, which we get from the software, and also a short sequence that is complementary to this scaffold part. So these 2 DNA oligos will undergo elongation in PCR single cyclers because they do 5 to 3' elongation, and this one 5 to 3' elongation. Then you get double-stranded DNA template, which can be used for in vitro transcription to get a single guide RNA, which starts after this T7 promoter sequence, which has your target sequence, then this scaffold sequence. So now you have guide RNAs, and we mix our guide RNA with Cas9, either mRNA or protein. So normally they both work well, okay? So we mix them together to do microinjection and a 1-cell stage embryo. Then we culture them to later stages, blatosis. So this is microinjection, culture to blatosis. Then before embryo transfer, we first do validation in embryos, meaning we do individual embryo lysis, and then we use primers that flag this double-stranded DNA break region. So this is control, meaning no guide RNA or no Cas9. So there's just one clean band. And for those embryos with Cas9 and single guide RNA, we see lower band or upper band, meaning there was random deletion or insertion. which suggests your guide RNA and Cas9, they both worked. Okay, so when we see this one, meaning after the validation, we will do one more embryo collection and a micro injection. And then we do culture to blastocyst stage, then we do non-surgical embryo transfer. So through cervix into uterine horn. Then after the birth, when we win these pups, we will collect ear tissues and then do ear PCR. And normally you should see these random insertion or deletions again, meaning their meaning CRISPR-Cas9 worked, but we can't send these PCR products directly for sequencing because this is a mixture. If you send these PCR amplicons for single sequencing, you will see these mixed results, which is not good for analysis. So of course you can do gel cutting or gel extraction, right? But sometimes for these ones, they're so close or there's kind of a smear, it's hard to do gel cutting and extraction. So what we do is TA cloning. We use these PCR amplicons and then use T vector to do TA cloning, because by doing this method, only one PCR product, either from this band or the middle band or the lower band, but only one PCR product will be aligned and sealed with this T vector. Then this T vector will be transformed into the E. coli. Then we do RB agar plate culture overnight. We will get many single colonies. Then we do single colony PCR. In each PCR reaction, you will just get the amplicons from either this band or this band or this band. So meaning, so these PCR products will be pure. Now you can send out for single sequencing. And you will get different results from different pups. So each one meaning one pup. Okay, so for example, this one means compare with this top reference. So the top is our reference. There's no change in all single clone PCRs, meaning this mouse is a wild type mouse. Either there was no DNA break, no repair, or there was DNA break, but the cell or the embryo used the homology directed repair to repair that, meaning there's no error. So there's no change. So that's why it's a wild type, okay? Or you can see this one has wild type allele. There's no difference as the top reference. But there's another mutant allele which has only one T insertion, or deletion allele, wild type allele. Or sometimes you even see this pup has no wild type allele, meaning this mouse is a homozygous knockout. But again, just want to mention this is a ear sample PCR, meaning at least in ear tissue, there's no wild type allele there. So this is a homozygous deletion. Or sometimes you also see mosaic founders, meaning in this mouse, you see more than one mutant allele. For example, you see C insertion and then some deletion. So this is one type of mutant allele, and another mutant allele, which just short deletion. So this means mosaicism, or this founder is mosaic in the ear tissue, because we do ear clipping lysis PCR. Okay, so that means the ear tissue is mosaic. We don't know the germline, because we cannot sacrifice this mouse to get the germline information. So what we can do is to wait this founder to grow out, to grow into adult. Then we do breeding with a wild type. And we don't think mosaicism is a problem for making mouse colonies. Why? Because sometimes after the breeding in the pups, in the next generation, we even see two different mutant alleles in different pups, meaning you will get this wild type mice, they have wild type allele from both this founder and this wild type breeder. And for this one, you wild type allele from this wild type breeder, but this mutant allele from this founder. And in another different pup, this one carries a different mutant allele. So what does this mean? This means in this founder, two different mutant alleles both exist in its germline, either ovary or testes. So that means one founder can give you two or sometimes even more mouse colonies. Okay, so this is mosaicism. But after breeding, if you always see just wild type mice or just heterozygous mice carrying only one mutant allele, just this one, for several litters, that means this mutant allele doesn't exist in its germline. Okay, even though the year has this mutant allele, but its germline doesn't have this mutant allele because this mouse is a mosaic. Different cell populations have different mutant alleles. So this is a mosaicism. Okay, so now I just want to quick summarize or compare the last two methods, which is embryonic stem cell mediated chimera and CRISPR-Cas9. Okay, because this method, you need to get embryonic stem cells, then you do chimera, then you do germline validation, then you do back crossing to get a pure genetic background. So that's why it takes much longer time, especially for the back crossing process. But a CRISPR-Cas9, you don't need embryonic stem cells. All the manipulation is directly done in embryos, and these embryos will be embryo transferred into recipients for animals. So that's why this method is straightforward, and there's no need to do back crossing because you will get animals directly. The background you used for your micro injection, the animal you get will also be that genetic background. Okay. And that's why it's faster, it's more convenient. And also because CRISPR-Cas9 doesn't need embryonic stem cells. So that means for other species who don't have embryonic stem cells. So far only mice and rats, they have authentic embryonic stem cells, meaning for other species, if you want to make knockout or knock-in, you can't use chimera method. You either do gene modification in somatic cells, then you do nuclear transfer cloning. That's before, okay? But now, most labs, they use large animals as the model. They do CRISPR-Cas9 because as long as you can get embryos from this species, you can do CRISPR-Cas9 directly in their embryos, and then you do embryo transfer. Okay, the only limitation is for large cassette knock-in. The CRISPR-Cas9 efficiency is still lower than ES cell chimera method, okay? And there could be some off-target issues. So that's why when you design the guide RNA, you should be really careful. Mosaicism, as I mentioned, if you wanted to just establish the mouse colonies, I don't think mosaicism is a problem. Actually, sometimes you even get more colonies just from one mosaic founder, so it's even better, okay? This is the main summary. So if you want to make a general knockout or short knock-in, for example, point mutation or short tag knock-in, CRISPR-Cas9 will be your top option. But if you want to knock in really large, complicated cassette, so CRISPR-Cas9 is efficiency still really low for large knock-in. So there's a high possibility of not working well with this method, meaning it's really challenging to do that in CRISPR-Cas9 system. So you may want to switch to ES cell electroporation, then select positive ES cells to do chimera. Okay. So far we have made 5 knockout and 1 knock-in mouse lines for these PIs, especially for recent large knock-in, which is a GFP reporter knock-in. It's a large knock-in, and it's successful. So it's really good. We are happy with this result. And this is an example of a knockout, which has been published already, so I can share here. So in this study, we target exon 1. So you can see, again, it's close to the beginning of the coding, protein coding part. So we put a guide RNA here, and after the deletion, there is frame shift mutation, meaning all the folding amino acid will be scrambled, will be wrong. And also we get a premature stop codon, meaning both the folding sequences will be wrong, and also it will stop much early, meaning really short is a truncated protein. So this is a successful knockout model. We also provide IVF surveys. For example, if you know, a collaborator has this mouse line and they have the cryopreserved sperm. Okay, so the sperm can be transported in liquid nitrogen. So we can thaw them, we can do IVF, embryo culture, and the embryo transfer. Or you can purchase them directly from the vendor. But again, just want to mention, you need to get approval from Animal Care Service and I'll cook first, because we need to make sure the vendor or the sperm, they are from the clean mice or clean facility. We don't want to get, you know, like any other dirty things here. Here's the video showing mouse in vitro fertilization. So after IVF, those fertilized oocytes will undergo early embryo development. Then we collect them, we do embryo transfer to get the pups. So far we have made 2 IVF lines for Tremblay lab. We also provide other services. For example, we did a Telvin IP and a retroorbital injection for Minter Lab to establish the mouse models. And here's one more example showing the model of wound biofilm, which has been used in Rotello Lab for comparing different nanoparticles. So we can provide the training to your lab member, so which will be good because it can save both the time and the cost for your project. So I will stop here. So with all of this, I want to thank our team, Animal Care Service team, and also all customers who give us the opportunities to work with this project. Feel free to ask your questions either directly here, or you can email me. Here the information. Thank you for coming, and thank you for your questions if you have.
Hi, Wei. Hi. So can I ask a question? Hi.
Sure, please.
Hi. Okay, great. Hi, thanks for the talk. And so we haven't done any in vivo animal model yet. Mm-hmm. And the reason I'm coming here is we are right now writing a grant, and we are thinking about if we want to include animal model in the grant or not. And so the question is like we try to do some of the knock-in, and maybe the target is around 1,000 base pair. And so it looks like based on the chart you show, it will take around 8 to 10 months for a successful knock-in, or is that the first of all, is that the time you are estimating here?
Yeah, I think the timeline works for most knock-in projects, meaning your 8 to 10 or even 1 year prediction is correct, because knock-in first, we need more validations during the process. We first needed to validate the guide RNA. So meaning we do guide RNA and a Cas9 microinjection to see if there is a random insertion or deletion. When we confirm this, then we will tell you, or we will tell you to design or to synthesize the template. Normally for large knock-in, single-stranded DNA strand shows a higher knock-in efficiency. And for around 1 or 2,000 nucleotide, so it's a single-stranded. So either IDT or GeneWiz or other companies can do this commercial service to synthesize this single-stranded DNA. So this takes around 1 month. When we get this, because only when we confirm which guideline is good, then we know in this region we will put this template. So that's why you need to make sure guideline is good. Then depending on the sequence around this guideline here, you will synthesize that specific knock-in template. And after 1 month, when this single-stranded DNA template is ready from the company, we will test if this template, knock-in template, works well in early embryos. If everything works well, then we do 1 more large-scale microinjection, then we do embryo transfer. Then we do, after 3 weeks gestation, then 3 weeks weaning age, then we can do ear clipping to see the genotyping, and then do sequencing or PCR. And because knock-in efficiency is much, much lower than knockout, meaning we need to do more round of microinjection and embryo transfer. So that's why it takes a longer time, and also the price is higher than knockout, just because you need to do more validation, more injection, more embryo transfer, more genotyping. So that's knock-in. Knock-in for 1 kb generally I think is fine now. It's not like as perfect as like a knockout, but the efficiency is fine now. But if you do 5 kb, that probably like too risky. 1 kb knock-in like GFP, right under a specific like before stop codon, meaning you want to use this gene's promoter to show the expression of this gene or something like for this strategy. 1 kb I think generally works fine now for CRISPR.
I see. Right.
Yeah. So
got it. Can I ask a follow-up question? And so in terms of the real price, and because right now targeting just R21, which is a really small grant, and so if we knock in 1,000 and gene, right? So what will be the estimated cost for that? Right?
So the cost, so knock-in generally is more expensive than knockout, but within knock-in category, there's no like formulation. For example, 1 kb knock-in is less expensive than 2 kb knock-in. There's no that, but generally knock-in will be, it's hard to say, but I think generally between 7 to 11 or 12,000, meaning depending on if in the first round everything works like smoothly, right? It will be maybe just 7,000, 7,000. But if it's like a really challenging one, meaning around that locus, knock-in efficiency really low. You need like multiple times repeat or something, then the price will be high. So as far as I know, some core facilities, they don't do repeat, meaning they just try 1 time. They just give me the template. I just do guideline, Cas9, your template, whatever you get, I just give the pups to you. In this way, the price will be low because they just do once, no matter you get that or not. They don't care. So that will be, the price will be lower. But if you just want to get your knock-in, if this is your final goal, so we would just like, we prefer doing validations at each step just to make sure finally, I think probably you will get your knock-in, but not like fail in the middle or something. Yeah.
Great. Yeah, thank you.
Yeah, by the way, just want to mention, you know, like for some commercial like vendors, the price is even higher because just because, you know, that is real business, but here our core facility is not like making like money. We just want to cover the revenue, or we just want to help the PIs. So that cost basically just cover, you know, like a guideline A, cover the mice, cover the per diem, cover our time or something. We are not like make money for that. We are just cover the cost. Okay.
Yeah, sure. Right. Yeah, I think that makes sense. Right. Thank you. Right.
Sure.
Are there any other questions? Okay, then I guess we're all set. Thank you, Wei. That was really informative. And thank you all for coming. We are planning for the fall seminars, and your feedback on this seminar series is very important. If there are cores or specific technologies you would like for us to feature, whether they are part of the centralized cores or not, please let us know. Also, we'll be hosting an in-person event in the fall, so please keep an eye out for additional information, and we look forward to seeing you. Enjoy your summer.
Bye-bye, guys.